Good afternoon, good evening, whenever you're watching this. This is my third attempt at this video because the first two times, when I got all done with the 20-minute recording, there was no video, just audio, which really wouldn't work out too well for you guys. So hopefully this one works. Um, we've already covered in class the Krebs cycle, which comes before the electron transport chain. Then you watch the video about glycolysis prior to that. So the focus today is going to be on the electron transport chain and oxidative phosphorylation, this last stage of cellular respiration, right? If you remember, the Krebs cycle completes the oxidation of our fuel, whatever that would be. In this case, we're focusing on glucose as our fuel, but as you know, you eat a lot more than glucose. So we'll take a look later at what happens with other fuel molecules that you eat, other organic molecules that you eat. So remember here, the point of all of this is to make ATP. And if we look at it so far through glycolysis, we've only gotten 2 ATP and Krebs cycle also just 2 ATP. Both glycolysis and the Krebs cycle use that substrate level phosphorylation. However, we're going to need a lot more than 4 ATP per glucose molecule that we consume to keep our bodies running because like a working muscle requires over 10 million ATP molecules per second. So 4 ATP out of a molecule of glucose just is not going to do a whole lot for us. So there has to be a better way. And there is. We can use what's called the electron transport chain, which is a bunch of proteins built into the inner mitochondrial membrane. If you remember, mitochondria have a double membrane, right? They have an outer and an inner membrane. The electron transport chain is a bunch of proteins built into the inner mitochondrial membrane. And you can see it in this picture here. We're seeing the three proteins built into the, or three plus some proteins built into the inner mitochondrial matrix. There's actually several proteins making up each one of these complexes here. So these are found within the cristae, which are the folds of the inner membrane, and the inner membrane is folded, remember, to increase surface area, so we can put more of these copies of the electron transport chain within the mitochondria. So we see transport proteins and enzymes making up this electron transport chain, and what they do is transport electrons down this electron transport chain from one protein to the next to the next. And that flow of electrons is linked to pumping protons into the space between the inner membrane and the outer membrane called the intermembrane space. And that's going to set up a proton gradient that the mitochondria are going to use to make more ATP. And when the electron transport chain is all done, we're going to have made about 38 ATP from one glucose. So we've already made 4. The electron transport chain is going to make 34 more. Okay. So remember, this electron transport chain only takes place if oxygen is present. So when we talk about the mitochondria, remember it has a double membrane, right? It's got the outer membrane and the inner membrane. The inner membrane's all folded up into cristae. And within the cristae are those enzymes and transport proteins of the electron transport chain. And this area between the outer membrane and this inner membrane is the intermembrane space that's filled with fluid that plays a really important role in the electron transport chain. So we're going to zoom in on one little section of the inner membrane of the mitochondria to look at one electron transport chain, but there are copies of the electron transport chain all along the inner membrane of the mitochondria. There's going to be thousands of copies of the electron transport chain. We're just going to look at one of those copies here, right? And so first, we're just kind of going to get set up where we're at within the mitochondria. This is the outer membrane. Here's the folded up inner membrane. And then here are your proteins of your electron transport chain. And so you get an idea. Here's the inner membrane. This is the intermembrane space here between the outer membrane and this inner membrane. And then this is the mitochondrial matrix down here. And then each of these proteins has a different name. You are not going to be responsible for knowing the names of these different complexes of proteins. This first one should tell you a little bit about what it does. So like if you saw the name, you would be able to figure out that this enzyme is going to be removing hydrogens from NADH But again, I don't expect you to know the names of all of these different protein complexes. Okay. So, if you remember from glycolysis and the oxidation of pyruvate in the Krebs cycle, we generated a whole bunch of electron carriers, right? We generated NADH and FADH2, and we said they had a bunch of stored energy. And now we're going to crack into that stored energy and use it to help us make ATP. So what happens here in the electron transport chain, right? Now we're kind of set up at where it occurs. Now let's take a look at what occurs. So we have these electron carriers that are going to pass electrons and protons to the electron transport chain. So we have our NADH and FADH2 that have their hydrogen that they're transporting removed from them. Okay, and then each hydrogen atom is made of one electron and one proton, and the electrons are stripped from those atoms, right? So what you're left with is a proton that's left behind. Okay, so the electron gets, so this NADH, it has its hydrogen removed. The electron gets stripped away and passed along the electron transport chain, and the protons, meanwhile, stay here in the mitochondrial matrix. As those electrons move from one protein carrier to the next in the electron transport chain, they are going to be producing energy as they are flowing, right? And why is there energy released from the flowing of these electrons? Well, every time these electrons move, they move from a less electronegative molecule to a more electronegative molecule, to a more electronegative molecule, to a more electronegative molecule. So every time they move, they're moving also to then a more stable configuration as they're moving to a more electronegative molecule. And since these electrons are moving from a less stable configuration to a more stable configuration, they release little bits of energy each time they move. And notice here the final electron acceptor in the electron transport chain after these electrons have moved all the way through is an oxygen molecule, right? And the oxygen picks up the electrons from the electron transport chain and combines with protons in the mitochondrial matrix to form water. So this oxygen is reduced as it oxidizes that final electron, the final protein in the electron transport chain. So oxygen is that final electron acceptor. So as these electrons move, the energy that they release is used to pump protons from the mitochondrial matrix into that intermembrane space. So we're building up this proton concentration gradient inside of that intermembrane space. We're getting an electrochemical gradient, or the intermembrane space is going to be more positive than the mitochondrial matrix. So we've got an electrical gradient, and then we have a concentration gradient of protons in that intermembrane space in higher concentration there than in the mitochondrial matrix. Okay, so the transport proteins in the membrane that are passing these electrons along are then pumping those protons into the intermembrane space. And that creates a gradient that is going to be harnessed to help make ATP. Okay, so to give you a visual, there is a hydrogen atom with one electron and one proton. And what happens is that is split into the electron and proton that make it up. The electron is then passed from one protein to the next, and as it passes, the energy is used to pump protons into the intermembrane space. Notice here that FADH2 drops off its electrons at a later point in the electron transport chain than NADH does. So as a result, fewer protons are pumped into that intermembrane space from the electrons that FADH2 drops off compared to the electrons that NADH drops off. And that little piece of information is going to be really important later when we take a look at how many ATP are generated from the electrons that NADH brings to the electron transport chain versus how many ATP are generated from the electrons that FADH2 brings to the electron transport chain. Okay. All right, so we're building up this proton gradient, which is super, super important, right? And like I said, oxygen is that final electron acceptor because it is the most electronegative element that we find in living things. And so it will draw those electrons from the last protein in our electron transport chain and pick them up. Okay, now this this energy that is used from these electrons to pump protons into this intermembrane space is then going to be harnessed to help add a phosphate group onto ADP to make ATP. So the phosphorylation that occurs as a result of the electron transport chain is referred to as oxidative phosphorylation, because the phosphorylation of ADP here is being driven by the energy from the electrons that were picked up from the oxidizing of our fuel molecules that we've taken in. The oxidation of glucose, in this case, has provided the energy to attach a phosphate group onto ADP to generate ATP. So in the electron transport chain, instead of substrate level phosphorylation, we're taking a phosphate group off of one molecule and adding it to ADP to make ATP. We're adding phosphate groups to ADP by harnessing the energy from the electrons that were stripped from our glucose or whatever other fuel molecule was used. Okay, so these electrons, like I said, are moving from one protein carrier molecule to another one, and each one along the way is slightly more electronegative. So what happens is we have a controlled oxidation, right? We are oxidizing one protein and reducing the next one, then oxidizing that protein and reducing the next one. And every time little bits of energy are released, instead of large amounts of energy all at once, if we had just taken those electrons and passed them directly to oxygen. We slowly release the energy from those electrons as they are passed from one protein to the next, and harness that to pump protons into that intermembrane space. So here we see the protons in the intermembrane space. Here's that inner mitochondrial membrane, and this is the enzyme ATP synthase. So we've set up a proton gradient, and now these protons basically have just one place that they can pass down their concentration gradient back into the mitochondrial matrix, and that is through this ATP synthase. There's a few exceptions to that. I'm going to have you do a reading about on a few of those exceptions. I'm going to talk about one exception in just a minute. But as those protons flow through ATP synthase, they spin a rotor within the protein that causes the ATP synthase to move into its active conformation, or its active shape. And as long as those protons keep flowing through, the ATP synthase stays active and synthesizes ATP. Okay, if protons are not flowing through ATP synthase, then it is in its inactive conformation or inactive shape, and it does not make ATP. All right, this concentration gradient of protons, by the way, is called the proton motive force. That's just what we call it. That's some terminology I would want to see you using properly in a free response question, is this proton motive force. So these protons diffusing down their concentration gradient is an example of what we call chemiosmosis, which is the diffusion of any ion across a membrane. In this case, it's protons. So we built up this proton motive force just so protons could flow through this ATP synthase enzyme down their concentration gradient to build ATP. Okay, so it is this chemiosmosis that links the electron transport chain to ATP synthesis, or it couples the electron transport chain with ATP synthesis, right? Because the electron transport chain was just used to pump protons into that intermembrane space. The energy from those electrons did not go directly to ATP synthase to allow it to make ATP. The energy from those electrons went directly to pumping protons into the intermembrane space, which then passed through ATP synthase to activate it so it could make ATP. So this chemiosmosis links the electron transport chain to ATP synthesis over here, or couples the electron transport chain with ATP synthesis. Okay, Peter Mitchell is the biologist who proposed the chemiosmotic hypothesis, which was really considered very revolutionary at the time. People thought he was a little out there, proposing that the movement of these protons could be harnessed to do work for the cell. But it turns out that it's not only in cellular respiration where chemiosmosis is used by cells to do work. We'll see other examples, and we've talked about a couple other examples already this year. Okay, and he proposed his hypothesis in 1961 and won the Nobel Prize for it in 1978. Now, One of the exceptions to those protons flowing through ATP synthase is something I'm going to talk about here, and I'm going to tie it into what's called thermoregulation, regulating body temperature, which is one of the topics that we need to cover in the course and exam description from the College Board. So this is a good place to throw it in there. It's kind of interesting here. Um, in cell respiration, decoupling oxidative phosphorylation from the electron transport chain is involved in thermoregulation, right? And that seems kind of hard maybe to understand here. Like, why would you not want to link the electron transport chain to making ATP, right? But let's take a look here at this example. This is used in hibernating mammals, which have a type of fat called brown fat, which most mammals have. It's brown because it's high in iron-rich mitochondria, and the iron gives the fat that brown color. Because if you think about like iron oxide, which is rust, it has that brownish red color. Brown fat has a lot of iron in it, which gives it that color. And that brown fat in most mammals has an electron transport chain uncoupling protein. And we'll take a look on the next slide at exactly what that protein is, but it's a protein that uncouples the electron transport chain from ATP synthesis. So this protein becomes active during hibernation. We'll take a look at how that happens also. And what this protein does is it allows protons to flow back down their concentration gradient through it instead of through ATP synthase. So no ATP is made. So the oxidation of the stored fuel, stored organic molecules that these hibernating mammals have still continues. So glycolysis and the Krebs cycle still continue, and so we still get this generation of this proton gradient, but the gradient that's generated is not used then to make ATP. All right, so we have the ongoing oxidation of the stored fuel. And what that does is it generates heat, and that helps to keep the body temperature up, right? Because none of the reactions that occur during cellular respiration are 100% efficient. A lot of heat energy is generated during those reactions as well. And so this helps to keep the body temperature warmer than the environment. And this method of keeping the body temperature warm is referred to as non-shivering thermogenesis. Thermo refers to heat, genesis is to create, right? So we're creating heat, and it's done without shivering, right? If you and I get cold, we start to shiver, and the movement of our muscles helps to generate heat to get our body temperature back up. But these hibernating mammals don't have to shiver to get their body temperature back up. They use this instead. And the reason why the uncoupling occurs is because if we get high levels of ATP, that ATP feeds back and shuts down cellular respiration. So then you also would not get the heat generated, right? So we don't want to make ATP this way when these mammals are trying to keep their body temperature up. So this is how this actually works, right? Here's a receptor in the membrane this is the membrane, and this is the receptor of of one of our brown fat cells in a hibernating mammal. And a hormone that these hibernating mammals produce called norepinephrine binds to that receptor, which is a G protein linked receptor. And when norepinephrine binds to that receptor, it activates a G protein that activates adenylyl cyclase, which is also sometimes called adenylate cyclase, same thing. So this adenylyl cyclase then converts ATP into cyclic AMP, which serves as a second messenger to activate a protein kinase, which then phosphorylates an enzyme in the cell it is a hormone sensitive lipase that becomes active when it's phosphorylated and starts to hydrolyze triglycerides, which are our fat molecules made of glycerol and 3 fatty acids. So when the triglycerides are getting broken down, the presence of those fatty acids activates the protein in the inner mitochondrial membrane called thermogenin. This is that protein involved in creating heat. So that name makes sense. When this thermogenin protein is activated, it allows protons to diffuse down through it. So these protons pass through thermogenin instead of through the ATP synthase molecule. So no ATP is generated from their diffusion, and all the reactions of cellular respiration have just been used to generate heat instead of ATP. So this has uncoupled the electron transport chain from ATP synthase, because the electron transport chain was still used to create this proton motive force, this concentration gradient of protons, but now they're not moving through ATP synthase. So the electron transport chain is not linked with ATP synthase or ATP synthesis anymore. Okay, so with cellular respiration, a quick little accounting here. Okay, we remember glycolysis made 2 ATP through substrate level phosphorylation, as did the Krebs cycle. And we said the electron transport chain made 34 ATP. Now, how can that be? How do we know it was about 34? Well, we know for each NADH that gets to the electron transport chain, the electrons that it drops off are responsible for moving enough protons into the intermembrane space to generate about 3 ATP. So for every electron, for every NADH that drops off its electrons, about 10 protons are pumped into the intermembrane space, which leads to the production of about 3 ATP per NADH. Because FADH2 drops off its electrons at a later point in the electron transport chain, enough protons are pumped into the intermembrane space to generate only about 2 ATP per FADH2 that's produced. So if we look here, glycolysis generates 2 NADH at 3 ATP each. That's 6 ATP from the NADH of glycolysis, another 6 ATP from the 2 NADH of the oxidation of pyruvate. Now for each NADH in the Krebs cycle with 3 ATP, we're going to end up with 18 ATP being produced from the 6 NADH of the Krebs cycle. And then the FADH2 that each produce 2 ATP in the end, we get 4 more here. So we get 6 plus 6 ATP is 12 ATP, plus another 18 makes 30 ATP, plus another 4 makes about 34 ATP. Okay. For a total of 38. So in cellular respiration, where did the glucose come from? We ate it, right? Where did the oxygen come from? We breathed it in. Where did the carbon dioxide come from? The oxidation of glucose, right? When we broke it down. Where does the CO2 go? Well, it goes from the mitochondria to our cell, from our cell cytoplasm to our bloodstream. From our bloodstream, it gets carried to our lungs and dropped off in our lungs, and then we exhale it, right? That's where carbon dioxide ends up. So all the carbon dioxide you exhale has been produced through cellular respiration. Where'd the water come from? It came from the reduction of oxygen, picking up those electrons as the final electron acceptor. Where'd the ATP come from? It came from substrate level phosphorylation and glycolysis in the Krebs cycle, and oxidative phosphorylation in the electron with the electron transport chain. What else is produced that's not listed? Heat, right? Which we just talked about, even more heat production with hibernating mammals. And why do we breathe? To bring in oxygen for cellular respiration, to get rid of the carbon dioxide that is generated from cellular respiration. So one last little interesting note, remember oxygen, that final electron acceptor, what happens if it's unavailable? Well, then the electron transport chain backs up because we don't have anything to pull the electrons down out of the electron transport chain, right? So NADH and FADH2 then cannot unload their hydrogens, right? And they can't be recycled back to glycolysis and the Krebs cycle. So ATP production is going to stop, which means that the cell's going to run out of energy. And you know what happens when it runs out of energy? It's going to reach metabolic equilibrium. And then you will reach at metabolic equilibrium and you will die. Cyanide is actually a poison that blocks oxygen from being that final electron acceptor. It will not allow the electrons to pass from the electron transport chain to oxygen. And that is why that ends up leading to death of individuals who take cyanide. And that's all I have on the electron transport chain. If you have any questions, please ask them during our review session, or sign up for an academic support time and we can go through a little bit more. So hopefully this was helpful for you, and I will see you guys in class.